I am Kalyani Pathode from B.Pharm third year. Now we are discussing the technique called as touch DNA revolutionizing. Touch DNA is the advanced technique which is widely employed in modern criminal justice system in many developed countries. So the aim of this technique is to extract genetic information from the biological substance. The methods involves recovering trace amount of DNA from the biological substance which is released by the released during the contact. So, the reliable DNA profile are produced by using the sophisticated kits, tools or equipped forensic laboratories. So, touch DNA is possible to be found various rights including the clothing, weapons and doors and handles from this area. So, what is forensic science? Forensic science is a multidisciplinary field in which forensic scientists used the methods like DNA analysis, fingerprinting, ballisticity and toxicology to produce the analytical information from the criminal intelligence. So, what is the goal of this forensic science is to uncover the truth and identify the perpetrators and making relationship between the father or paternity relationships can be found out by forensic sciences. So the modes of the touch DNA sampling is by swabbing testing techniques, tape lifting and cuttings or scraping. So also the touch DNA extractions can be carried out by which the touch sensation can be extracted all the information from this extracting process. So organic extraction also can be carried out by organic solvents are used like ether, benzene, etc. Also automated DNA extraction can be carried out. Now we are also identified by the amplification process. Amplifications is a process in which extraction of DNA followed from the applicon plays crucial role in DNA profiling and this process. Then second steps are by capillary electrophoresis. Electrophoresis in which the movement of DNA or in which multiplication can be carried out. After amplification, DNA fragments are separated by using the capillary electrophoresis. The data obtained from the electrophoresis are analyzed using the given MAPEX IDX software. Then some challenges can be also carried out in this for the sampling. So the handling will be not proper if the extracted DNA can be contaminated or degraded at that time. So to collect it very properly and handling will be sure to be maintained by forensic scientist. So the forensic science which enhance the assessment of evidence. Also maybe the scientists have enhanced the during collection, analyzed and process. In that process the 90% of DNA will be lost. So how we can manage the 10% of DNA from the crime scenes? So it collect properly by the virtual techniques. So the workflow is crucial for potentials in improvement and efficiency of all the types of biological samples, just like a semen, blood or saliva. The successful of touch DNA forensic tools which depends on the optimizing technique with sample collection and to profile generation. Then the benefits of some of the benefits of this technique is as follow is first is specificity. If the small quantity of DNA is required in that time since then you can by multiplication or amplification process you can collect the more of the certain amount of DNA or RNA can be takes place. Then it is very fast and effective process and it takes less time duration. So also it versatility also available in which if small surface area can be collected the samples also taken place. Also if technique is very advanced. There are some limitations of that techniques are first degradation, sample destructions or contaminations can be carried out by this technique. So now we also identify these techniques by DNA profiling in which unique genetic fingerprints are represented and the individual genetic makeup. So thank you for this delivering the video.